Okay, perfect. We should be all set there. How have you been, Cindy?
Good. I don't often leave my house, but other than that, good.
Me too. I kind of love it.
At first, and everybody was really upset. I wasn't really at all because my introvertedness was just fine, finally
Right?
taking a breath and hanging out. But yeah, I'm ready to get back into things as soon as the world is ready.
Yeah, I think we're all breathing a collective sigh of relief that vaccine stuff is moving forward and right.
Cautiously optimistic.
Cautiously optimistic. My sister lives in Missouri and she got a notification saying, hey, it's going to be widely available in 4 weeks from now. And, from somebody official in the government. And I said to her, I'm like, I don't know if that's right.
Yeah, I think the scale up will probably take longer than that, but I guess we'll see. 4 weeks would be pretty optimistic. I mean, even to do like the safety testing for storage and things like that, I would imagine that's take a little bit longer. But again, cautiously optimistic.
Exactly. Yeah, I am hopeful, but wait and see, right? Yeah. I'm in the same boat where I've been doing just fine, but I'm finally starting to get stir crazy. Which, it took 8 months, but starting to get there.
Yeah. Well, now with winter setting in too, I think it's going to get a little bit harder.
Yeah. Yes, but at least we have multiple reasons for optimism now. So it'll be an easier winter in some ways than we thought it was going to be. I'm not able to say that the glass isn't half full, but it's like got some liquid in it.
Yeah, a little bit of optimism is a good thing. This is a great audience. Boy, you guys have really recruited from around the world and back again.
Yeah, people are here where it's the middle of the night for them. So props to everyone showing up from all over the place. So maybe we'll just give it like one more minute to let people keep trickling in. But yeah, this has been a lot of fun. We've been so happy that people have been, great people are agreeing to speak, and also people are showing up, and it's been a really fun community.
Well, honestly, I mean, I think at least for me, that's one of the benefits. I don't really miss travel so much because I was doing it at such a feverish pace there for a while. So things like this is so easy to say yes to because you don't have to leave the comfort of your own home.
Right. Don't have to get on a plane and spend some exhausting days traveling. Yeah, it's been good. Although I do miss real conferences.
And people interaction for sure.
Yeah. All right, let's go ahead and get started so that you have enough time. So hello to everybody, welcome as usual. We had a 1 week break for the madness that was the last week, but we are back now at Cell Migration Seminars, and we're really excited today to have Cindy Reinhart-King. So Dr. Reinhart-King is the Cornelius Vanderbilt Professor of Engineering and the Director of Graduate Studies in the Department of Biomedical Engineering at Vanderbilt University. Prior to joining Vanderbilt in 2017, she was on the faculty of Cornell University. She received tenure there in the Department of Bioengineering. And before that, she'd obtained her undergraduate degrees from MIT and her PhD from the University of Pennsylvania. So Dr. Reinhart-King's research interests are in the areas of cell mechanics and cell migration, specifically in the context of cancer and atherosclerosis. And so she's been awarded a ton of different awards, just a few of them to mention from the Biomedical Engineering Society. She has both the Rita Schaefer Young Investigator Award and also the Mid-Career Award. And then she's won numerous awards involving diversity, contributions towards improving climate for women, and the Vanderbilt Chancellor's Award for Research. So she's also a fellow in the Biomedical Engineering Society, American Institute for Medical and Biomedical Engineering, a bunch of other different positions that I won't go through right now, but basically She's pretty great, and we're really excited to have her here and hear about some of the recent research from her lab. So thanks so much for being here, and take it away.
All right. Well, thanks for the chance to present. I mean, this is an amazing forum and a terrific audience. I think, with the exception of maybe Gordon research conferences, I've never spoken to an audience that's so right in my research area before. So this is nice. I can try and speed through or even skip all that sort of intro of why cell migration is important, right? Why are my slides not advancing? There we go. Okay, so my lab is broadly interested in cell mechanics, so cells exerting forces and forces getting exerted on cells. And we basically work in 3 areas: atherosclerosis, metastasis, and angiogenesis. Today, I'm really going to focus mostly on the stuff that we've been doing in metastasis and single cell and collective migration. And then I'll dovetail that into some of the work that we've done in angiogenesis. Okay, so we're biomedical engineers, and most of the folks who join my lab, what I try and train them to think about is taking a lot of what we know about single cell behaviors and scaling it up and thinking about it in the context of human pathophysiology. So usually our general approach is we start with a lot of ex vivo characterization of tissues, usually human or mouse, and that includes things like AFM and histology and optical imaging to look at mostly ECM characteristics. So mechanical properties, architecture, structure, fiber size, things like this. And then we use a lot of different tissue engineering approaches and biomaterials and microfabrication to try and rebuild those characteristics that we identify so that we can understand how the cells behave, so that we can understand how the molecules within the cell control those behaviors, with the idea of identifying therapeutic targets. Okay, so I think this audience is pretty familiar with the hallmarks of cancer. There's this big paper written by Bob Weinberg in 2000. I think it's the most cited paper in Cell ever, at least the last I checked. And basically what they did was they defined the hallmarks of cancer that basically categorize a tissue as malignant or not. And it includes a lot of things that we talk about often: angiogenesis, uncontrolled replication, sustained proliferative signaling, things like this. So this was in 2000. In 2011, they came up with an updated version of this paper, and they added several hallmarks, including deregulation of cellular energetics. There are a few others, including avoiding immune destruction. So this timed with the advent of things like immunoengineering and immunotherapies, and inflammation, which is really a hot area right now, and looking at macrophage infiltration and things like this. But the one that caught our eye was this deregulation of cellular energetics. And so when you think about the hallmarks of cancer, actually, so these are the original hallmarks in here, you can take almost each and every one of them and relate it back to cellular energetics or metabolism. And so you can think about things like tissue invasion and metastasis, which we know is regulated by HIF-1 alpha, which is induced in hypoxic environments. And HIF-1 alpha also has been linked to glycolysis. P53, which is the most mutated gene in I think 70% of cancers, you can link back to glycolysis, and so on and so forth. So almost every aspect of cancer you can link back to cellular energetics in one way or another. So how's energy actually used in a cell? Why do we care about cellular energetics? There's a few obvious things. This chart came out of a really good book. If you haven't seen it, I highly recommend it, by Phillips and Milo, Rob Phillips at Caltech, Cell Biology by the Numbers, where they go on to basically try and quantify different biological processes, basically setting numbers to things, pulling out of the literature and trying to do various calculations of fundamental biological processes, including energetics. So how's energy actually used? So things like protein synthesis for proliferation is obviously a big one. In addition, when you think about the heart and cardiac contraction, ATPases and actin-myosin contractility is a big one. And so we started thinking a little bit about this in the context of cytoskeleton and cell migration, because we know ATP is necessary for actin-myosin interactions and contractility. And there was this paper that came out several years ago that actually tried to look at this mostly in the context of neurons. And so what they did was they blocked ATP synthesis, which is down here on the x-axis. And then they measured the rate at which ATP gets depleted. So basically the cell can't make ATP, and then they're just going to measure how quickly the ATP stores go down. And what they find in the control situation is that over time there's complete ATP depletion, right? So that you're not making any more ATP, you're just going to burn it. But if you inhibit various aspects of actin contractility, actin assembly, actin myosin interactions, you basically plateau at a much lower amount of ATP depletion, meaning you keep some amount of your ATP stores. And so what this was basically, why this study is important, is because it's really one of the first pieces of evidence to show that the cytoskeleton is responsible for a significant amount of the ATP burn in a cell. And again, in the field, when you think about energy utilization, usually people are thinking about it in terms of protein synthesis, they're thinking about it in terms of proliferation, but here we are thinking about it in terms of just cell architecture and cytoskeleton. Okay, so this led to our question, which, you know, honestly is a pretty fundamental question, but hadn't really been tackled too much before we started looking at it, which is how do cellular energy levels contribute to migration ability? And basically just simple questions. How much energy does it take for a cell to move? And again, sort of intuitively obvious, the cell has to contract or send out protrusions. It reorganizes matrix, it degrades matrix. Presumably these are all energy intensive activities. How much energy does it take? And I think, you know, for this audience, I would assume most of you would say, oh, of course, this seems like a pretty natural question to ask. But when we started this work about 6 years ago and started talking about it, most people said, sure, it takes energy, but really proliferation takes the most energy. And so the amount of energy it takes to migrate is pretty minimal. And so we should really just, of course, focus on proliferation. But that did not deter us because we study cell migration. So off we went. And we asked several questions. So first is, what's the relationship between migration and energy utilization? So we basically wanted to know whether or not the amount of energy that the cell could either make or utilize or burn affected its ability to migrate, and maybe even basically predicted how it could migrate. How does the terrain affect energetic needs during migration? So again, for this audience, not a unique idea that if a cell has to squeeze or degrade matrix or move over something soft or something stiff, presumably those are all going to require different amounts of energy. How does energetic needs actually affect migration decisions? So can the amount of energy a cell needs affect whether or not it's going to take one path versus another? And then what role do energetics play in collective migration? Okay, so my lab's been interested in the problem of migration for many years now. One of the things that we and others, of course, have noticed is that there's a lot of heterogeneity in cell behaviors. And so this is a mammary tumor. So we injected MDA and B231 cells orthotopically into the mammary stroma of a mouse. Then we took that whole tumor out after it grew, put it down on a microscope slide. The purple is collagen fibers, and the green, of course, are the cells. And what I'm trying to show you here basically is that over a certain time course, 48 hours here, what you find is that some cells really don't move at all, whereas other cells move quite fast. And all of these cells came from the same population. So why is it that some cells move well and others don't? And of course, this is a really complex environment. What if you just take cells and look at them on a petri dish? And I'm sure, again, everybody in the audience has done this. You'll find the same thing, that there are some cells that will zip across your field of view and others that don't really move at all. And so we've been thinking about this in the context of whether or not, or what determines the ability of cells to migrate. So when a cell hits an environment, what will determine whether or not it's going to get up and go? Is it stochastic, meaning it's going to just change in time, or is it deterministic, meaning there are just some cells that are more inherently fit? And then the second question is context-specific mechanisms. So in other words, can you tweak the environment to make it more conducive to migrate so that you could get a cell that normally wouldn't go anywhere and get it to migrate by giving it the right environment to do so? And this is incredibly relevant in vivo because, as we know, the environment is very heterogeneous in space and time. All right. So we've done a lot of work basically trying to recapitulate matrix architecture in a Petri dish. If you were to just buy collagen from Becton Dickinson or name your favorite collagen company and polymerize it into a 3D collagen matrix using the protocol they give you, you get something that looks like this. But this is actually what the collagen looks like inside a mammary tumor. So while I think you can stretch a little bit and say they're similar in that they have both fibrous architectures, in reality, when you look closely, a lot of the features are in fact very different. So you can find areas that are fairly dense, and so you can control the density of the collagen. You can find areas with really porous, large fibers, large pores. You can also find areas of alignment. So we've worked out different methods to align collagen. And then you can find really large spaces, these channels that exist, and we've worked out different microfabrication techniques to do that. Okay, so back to the energy question, using these different terrains to understand how cells navigate and how much energy they use to do so. One of the things that has really enabled us to work in this area are the advent of single cell biosensors. So if you were old school metabolism, you've been working in this field for a while, you know that most people look at metabolism by looking at whole cell populations using usually a technique called seahorse, which is going to tell you about the overall acidification of the media due to cells, the overall oxygen consumption rate, things like this. But a few years ago, various single cell biosensors started coming out, and we adopted one developed by Gary Yellen's lab, who's at Harvard, and it's a measurement of the ATP to ADP ratio within an individual cell. And so we can put this into cells. And basically in one wavelength, it'll fluoresce the ATP signal, and a second, it'll fluoresce ADP. You can put one over the next. The probe itself is called Percival HR, so it's the Percival HR signal that we're reading off. And basically what you find is that you can get this ratio of ATP to ADP. Now, when the Yellen lab developed this, they had used it mostly in bacteria. There hadn't been a ton of work done in mammalian cells. So we put this into MDA MB231 breast cancer cells. And we first wanted to make sure that it was working as we thought it was. And so we basically just put cells into increasing concentrations of glucose and complete media. And what we found is that the signal, the Percival HR signal, goes up with glucose concentration, which makes sense. So you're giving the cells more glucose, they can make more ATP, and then you're going to see more ATP in the cells. Likewise, we also did the same thing with serum. You can give the cells more serum and you can get more ATP to ADP signal. Okay, so back to the question, what about how does this ATP signal correlate with the ability of cells to migrate? And again, the right audience to appreciate how painful these experiments were. My graduate student, Matt Zanatelli, basically what he did was he monitored individual cells over time. He looked at their stepwise speed, so their speed between any two frames in time, and then looked at their ATP to ADP ratio in that same time step. And then he did some cross-correlation analysis. So this is the measurement here. So here's the speed. The speed is very heterogeneous, which is what we'd expect from step to step, as is the ATP to ADP ratio. We then did this for many, many cells and derived out a correlation coefficient. And what we found is that they're in fact pretty highly correlated. So there's about a minute lag between when a cell burns ATP and when a cell takes a change in velocity forward. And so, so basically what this is telling us is indeed there is a relationship between ATP utilization and the ability of cells to move. Okay, so what if we change the terrain and we make it harder for cells to move? And so in this case, we increase the density of collagen, and what we found, these are our cells embedded in collagen. What we found is that as you increase the density of collagen, you increase the amount of ATP to ADP made in the cells. So basically what this suggested to us is that if you give the cells an environment that's harder to move in, we know they move slower in these denser collagen gels, they have to degrade more matrix, they can't slip through pores, they have to actually carve their own path, that they make more ATP to do so. But this result was a little bit confusing too, because you could interpret it a different way, which is that The cells have more energy at the 5 mg per mil gel because they're not using as much. So in other words, the cells aren't burning as much ATP, so they have larger stores of it. There's some data to suggest that cells don't really store ATP in that way, but that data was really hard to nail down, and so we wanted to figure it out for ourselves in this case. So what we did was we looked at the uptake of this probe to MBDG. 2-MBDG is a glucose analog, so basically it tells us how much glucose the cells are picking up. So as the density of the collagen increases, they pick up more glucose. So the cells have more ATP to ADP, and they're picking up more glucose to make that ATP to ADP. So basically what we've shown is that there's more input, more glucose, there's more of the actual product, ATP to ADP, and then we looked at burn. So this is ATP hydrolysis, and we see that also go up in more dense collagen gels. So basically what it's telling us, the cells are more metabolically active in stiffer, more dense collagen. They take in more glucose, they make more ATP, and they burn more ATP. Okay, what if we make the environment easier to move in? So in this case, we created aligned collagen gels. We do it by just pulling magnetic beads through. There's a few different ways to do it. That's our method that we've utilized. We quantified the amount of alignment here just to show that our fibers are aligned. We show here that the cells do move faster in aligned matrices than random. Again, none of this is new, right? This is all well established that if you give cells aligned collagen fibers, they tend to move quickly and very directionally. But then we looked at our ATP to ADP ratio, and what we see is that when they're on aligned fibers, which they typically move faster on and more easily, they have less ATP to ADP, they take in less glucose, and they burn less ATP. So basically, when the road is clear and it's easier to move, the cells don't need as much energy, they don't make as much ATP. Okay, so the next thing that we asked is whether or not, if we halted migration, whether or not we could drop that ATP to ADP ratio. So these are very similar to the experiments I showed you way at the outset that had been done by others looking at neurons in the context of basically freezing the cytoskeleton, but hadn't done it with this single cell level. And so we did it here. We used our ATP to ADP sensor. We inhibited all of our favorite contractility mechanisms, so Rho and myosin and actin polymerization. And basically what we find is that when you halt cytoskeletal activity and the ability of cells to migrate, you drop the ATP to ADP ratio, you drop the glucose uptake, and you drop the ATP burn. So basically, you make the cells less metabolically active. Okay. So basically what we've set forth so far is that there is a relationship between migration and energy utilization. The more energy a cell has, the better it seems to migrate. The terrain does affect energetic needs. So the easier the terrain is to move through, the less energy the cell makes. So the question that we went after next are whether or not the energetic needs affect migration decisions. So if a cell's given a path, one of which is quite challenging to go through and one of which isn't, which is the cell more likely to use, and is it related to its ability to generate energy? Okay, so the way we proceeded through this problem was we really focused on the use of microtracks. So if you look in vivo, what you can find actually are some fairly large interstitial spaces that exist in collagen. They can exist along nerves, they can exist along blood vessels, they can also exist due to the activity of leader cells, so cells that can actually move out of a tumor first and carve paths in the matrix. And so we wanted to come up with a technique to basically recapitulate these paths and the bifurcations in the paths. And so to do that, we developed a microfabrication platform a number of years ago. It's very similar to the microfabrication that people have used to stamp proteins and things like this. Only instead of using it to stamp proteins, we basically use it as a mold. So we create our master here and cast it into a PDMS stamp. It basically has long pillars, columnar pillars that on the PDMS stamp. We depress it into collagen as the collagen is polymerizing, and we pull it out. We then seed cells into these depressions. We wash away all the excess cells and cap it. And so what you get basically is a cell sitting in a channel that's fully encapsulated. So this is a Z view, so you can see it's on both, attaches to both sides of the channel, on the right and left, and here you can see attaches on both sides, top and bottom. Okay, what's a cell look like in these channels? It zips along. They move actually quite fast, and they move quite unidirectionally. We have a few papers exploring this and trying to understand why it is that they move so unidirectionally, which I won't go into here, but basically it's just a good way of confining a cell into a defined path. Really, the only time it turns around is here. It's hitting the end of the path, and here it hits this piece of debris, which causes it to flip directions. Okay, there we go. So the first thing that we did, remember our question is basically, how does energy affect the ability of cells to move in a given direction, was to define the boundary conditions for confinement. So basically, how big should our channels be that we allow the cells to move in? And so what we did was we started by creating a tapered microtrack, and it goes from fairly large to fairly narrow. We put cells inside of it, and then we watched them move. And we looked for two things. We looked for the width of the channel where the cell now contacts more than just one side. So you can imagine if you made this infinitely large, a cell is just going to attach to one side. So we waited for the cell to be able to touch both sides. And then we also looked at, when does a cell turn around? And so this is it. So here's our cell in our tapered track. Here's the cell on just one wall. It tends to elongate and look pretty mesenchymal. As it moves along one wall, it eventually gets to a point where it will crisscross and touch both sides of our channel. And then finally it'll eventually turn around when it gets too narrow for the cell to move. And so, so the point at which the cells, these particular cells, will touch both walls is about 12 microns. And the point at which they turn around is about 7 microns. The one thing I'll say that I think will make sense to those of you who are familiar with some of the work that people like Dennis Disher and Denny Wirtz are doing is, one thing we didn't look at here is nuclear deformations. So, so at 7 microns, we chose that, one, because that's the point at which the cells turn around, but two, we also don't see any large nuclear deformations as they move in that confined space. And so it's really a good spot for us because we're not inducing any of we don't think we're inducing any of the things that others have reported in terms of histone unraveling and changes in gene expression due to confinement and compression and things like that. Okay, so what we did was we used the microchannel patterning methodology to create tracks with a bifurcation. So this is our feeder track. Our cells start here. This is 15 microns. They head towards the bifurcation, and the bifurcation presents them with a 12 micron track. So this is a track where they can touch all the sides, or a 7 micron track, which is just at the uncomfortable limit for cells. So they'll move into it, but it's right where they often turn around. Okay, so we basically watch cells as they move along. Again, you guys who do cell tracking can appreciate how painful some of these experiments were for the student, Matt, doing them. He basically watched cells in the bifurcation. He watched them hit the bifurcation and then make a choice between wide or narrow. Okay, so the first thing he noticed is that as the cells go into the track, not surprisingly, if there's in a really wide track, the channel deformation is actually negative, meaning the cell pulls in on the track. Not surprising, that's what cells do. If the track then gets more narrow, 12 or 7, it actually pushes it out. So, so these are collagen, they are deformable. We did do experiments where we made the channels less deformable, which I won't really get into. But that is a key element here, that the cell can deform, but also the collagen matrix is deforming around the cells. But it indicates that the cell has to do a fair amount of pushing to get into that channel to basically open it up for itself. Okay, so what happens to the cells in these channels? They basically have exhibit 3 types of behaviors. So as they hit the bifurcation, they can either turn around, they can head to the smaller path, or they can head to the wider path. cells that start on the path that leads into the narrow channel. So this is our narrow channel here. This is the cell, basically the cell traveling along this wall and then heading up into the narrow path. And what happens is if the cell starts touching the wall, heading towards the narrow branch, not surprisingly, it typically heads into the narrow path. If, on the other hand, the cell starts on the side of the wall that leads into the wider branch, then it'll typically move into the wider branch. And that's the dark bars here. What's really interesting are the orange bars. So if the cell travels down this 15 micron path and is touching both walls as it hits the 7 or the 15, as it hits the bifurcation, the 7 or the 12, it's more likely to choose the 12 micron path. And this is basically what this is telling us. There's about 25% of the time it'll choose the narrow branch, about 70% of the time it'll choose the wide branch, and then some fraction of the time it'll turn around. But clearly the cells have a preference for choosing that wide branch. Okay, so our question, obviously, based on the rest of the talk I've given, is whether or not energetics play a role. And so that's what we measured. We looked back at the ATP using our Percival probe. And what we find is that as you increase the channel width, you decrease the amount of energy the cells need. So when the path becomes easier to move, the cells don't make as much ATP to navigate within it. Okay, so what does that mean in terms of actually choosing a path? So what we did to figure out whether or not energetics actually leads to choice of path, so to speak, is we tracked cells, their ATP to ADP ratio as they move down the feeder track and then as they make their choice. And then we grouped the cells based on whether or not they chose the 7 or they chose the 12. And what we found is that you can predict somewhat reliably a priori which direction the path that the cell is going to choose based on how much energy it had as it moved down the feeder track. So basically, if a cell was in the feeder track and had higher energy levels, it was going to be more likely to choose the 7 than if it had lower energy levels, where it's going to be more likely to choose the wider 12 path. So the amount of energy the cell has is going to predict the likelihood of it going into one path or another. All right, so that was most of the work that we had done, laying a foundation, looking basically at single cell behaviors. But again, for this crowd, as you know, the field is moving away from looking at just single cells and looking at a lot of the energetic dynamics and leader-follower behaviors. And so this is what we did. We were interested in leader-follower and specifically how much energy it takes to be a leader compared to how much energy it takes to be a follower. And I won't go into this because I think this crowd knows that the difference between single cell and collective migration, the fact that collective migration might actually be the more common mode for most cancer types. So there's strong motivation to understand it. Okay, so we were actually highly motivated by this problem by a paper that Bob Austin's group had published in 2013. And so this was really the first evidence we could find of someone suggesting the role of energy in collective migration. But basically what he did was he created a platform here that created a glucose gradient. So the cells started up here. They had to migrate through collagen towards a glucose gradient. And what he found is that as they do this, they were migrating collectively. But what he found is that if he watched them over really long periods of time, the organization of the cells would shift. And so in terms of the position of the leader and follower, after several days, the leader and follower would actually switch position, switch again, and switch again. And so he suggested in this paper, largely through thermodynamic calculations, that this shifting of the leader to follower cell was guided by energetics. So the idea he put forward was that the leader cell basically takes a lot of energy to be a leader. It takes a lot of energy to lead the collective strand and the followers. That it was the cell responsible for most of the matrix reorganization and leading the rest of the cohort. And that at some point the cell will burn its energy, it will drop back to be a follower, and a different cell will take over as the leader. But again, most of this was done through the observation that switching happens, and then just the calculations of the thermodynamics of the system. And so we wanted to explore this a little bit further. Now that we had these probes that could actually tell us about the energetics, we wanted to actually try and quantify it experimentally. So we use a spheroid system to induce collective migration. We've made tons of different types of spheroids. Here I show a mix of MCF10A and NDA MB231s. These were particularly intriguing to us because not surprisingly, the 10As that are non-malignant don't really migrate out into the surrounding matrix. Those that are malignant do. And then when you mix them, actually, you can get these strands that form where the normally non-migratory, non-malignant cells start playing the role of follower. And they follow along in these strands. And you can see that here. So the 231s are in green. They typically leave the spheroid first, and then our 10As will follow behind. We actually did this experiment with a bunch of different types of cells. So here was a metastatic breast cancer cell and a non-malignant breast cancer cell. This is a highly aggressive metastatic cell, and again, the non-malignant. These are prostate cancer cells. Highly metastatic prostate cancer, and then a non-malignant prostate cancer cell. And again, we saw basically the same thing, that the more metastatic cell becomes this leader. And so if you actually look at the collagen as the cells migrate out, what's intriguing is, first, I guess I'll go through the data that others have shown. So this isn't surprising right here. If you take non-malignant cells, embed them in collagen, they'll actually reorganize the collagen to bundle around it parallel to the spheroid. So it sort of tucks them in. And this is us quantifying it here. It's really highly aligned around the non-malignant spheroid. I'll skip down here. If you look at just the malignant cells that are highly migratory and move into the collagen pretty readily, they'll realign the collagen so it's more perpendicular to the spheroid. And people have reported this extensively in vivo as well, mostly Patty Keeley's group and Paolo Provenzano. And so this alignment here is thought to actually facilitate migration. Now, if you combine them, what you get is basically you can find situations where there are leader cells, the follower, the non-malignant cells behind. And if you zoom in, what you find is that these malignant cells are actually carving paths. So you can find basically these microchannels in the matrix, which the follower cells are moving through. So again, it speaks to this really important role of the leader in matrix reorganization, which we speculated was highly energy intensive. So when we looked carefully at our system, and these are actually spheroids of just 231 cells and high density collagen, what we find is if we look at our strand, which we've highlighted here, and we look at it in time, the follower cell here is in the white arrow, the leader cell is the red arrow. And if you look over really long time periods, so this is 450 minutes, what you can see is there becomes a point where the leader and follower switch places. And so this is basically what Bob has shown, Bob Austin's group has shown in his micro device. We've now shown happens in these collective strands. And the question that we wanted to ask is it dictated by energetics, knowing that that leader cell plays such a key role in reorganizing the matrix? So we looked at the glucose uptake. So this is our 2-MBDG probe that I mentioned earlier. That basically tells us how much glucose the cells are taking in. And what we find is that the leader cells, the tip cells in these strands, take in significantly more glucose than the follower cells. Okay. What's interesting is if you look at the leader lifetime, it says it is a function of the matrix density. So if you watch the cells migrate in these collective strands in varying densities collagen, what you find is that when the collagen is not dense, 1.5 mg per mil, and not very stiff, the leader cell tends to remain the leader cell for much longer. If you, on the other hand, on the other extreme, put the cells in really dense collagen where it's harder to move, the leader lifetime drops off a lot faster, and there's significantly more switching. So this is Kaplan-Meier analysis. So you've probably seen this for like patient survival, but we did it for leader cell survival. And again, the leader cells tend to fall off a lot faster when the matrix is harder to move through. So what if we now look at glucose uptake as a function of matrix density? So what we find is really two interesting results came out of this. The first is that as you increase the density of the matrix, you increase the amount of glucose the leader cell takes up. So it's harder to move in, the cell's going to take up more glucose. But what's interesting is that if you increase the density of the collagen and you look at the follower cell glucose uptake, it doesn't change. So it's really the leader cell that's bearing the burden of the changing extracellular environment, and the follower cells benefit from that. And their metabolic activity basically remains level regardless of the density of the matrix. Okay, so my conclusions for at least this part of the talk is we're really pursuing this idea that cancer cells use drafting. And so I'm sure you have seen this in one form or another. It's well known in geese that they flock in V's to help minimize wind resistance. It happens in NASCAR car racing. The reason they travel so close to each other is to try and minimize the wind resistance and maximize the benefit from the draft. If you're into cycling, traveling in a peloton, that the cyclists will work on as a team so that the follower cyclist can save energy. And so we think nature is using basically the same principle in cancer cell movement, that there's a way to do that to minimize energy. The direction we're taking this now, we've got a lot going on, but one of the questions that we're asking, but is definitely quite a struggle, is in each of these examples that I just showed you in geese, NASCAR, and cyclists, they communicate with each other to trade places. So the geese will actually honk at each other to trade places. Teams on NASCAR wearing headphones and can talk to their team managers, and even cyclists can talk to each other as they switch places. And what we're not clear on is whether or not cancer cells also have a way of communicating. So is it an active process where they switch places, or is it just that the leader cell gets depleted, it falls back and gets taken over? That we don't know. So a lot of interesting questions there. So the summary from this part of the talk is that migration is energy intensive, leading requires more energy than the following, and that metabolism, migration, and the ECM are linked. And I know the last one seems probably pretty obvious to this audience, and I think there's more coming out now in the past year, but when we started this work, most people told us to stick with proliferation with regard to metabolism. So hopefully as a cell migration audience, you can get a little bit excited about linking metabolism to this field. And why is it interesting? It's an easy connection to make translationally. So the first metabolic inhibitor for cancer came out in 2017, anastrozole. There's another one that just came out last year. And so we're only talking 3 years or so of going after metabolic targets for cancer. And so I think it's going to be a growing field, one that picks interest. And again, when we're talking about why these are prescribed, they're prescribed to halt proliferation. But what if they're also a mechanism to halt metastasis? So that's of our interest. Okay, so I have a few minutes. I'm going to tell you what I hope is sort of a quick story on translating some of this stuff to angiogenesis, and at the end I'll tie it together, I promise. So my lab's been interested in angiogenesis for a while. It was actually work that I started as a PhD student now 20 years ago. So we've been thinking about this problem for quite some time. And with our focus in cancer, of course, we're interested in what causes blood vessels to grow in tumors, but also not just what causes them to grow, but what causes them to grow in the architecture that they do. So when tumor angiogenesis happens, you don't typically get the well-organized vascular structures that you would in normal tissue. You tend to get pretty tortuous, very leaky blood vessels, which can cause all sorts of problems for creating tumor pressures and impaired drug delivery and things like this. And so one of the questions we've been asking is whether or not the extracellular environment in solid tumors, the fact that they're stiffer, does that stiffness actually contribute to the abnormal vessel structure? And so this work was actually motivated by a study. This was the very first paper my lab ever published, my independent lab. And it was basically just describing that if you take endothelial cells, this is in the absence of any sort of angiogenic cocktails or anything like that. You just take endothelial cells, you put them on a soft substrate, and they tend to self-assemble into networks. And if you put them on a stiff substrate, they tend to just act as individual single cells. And so basically it told us that these compliant substrates promote cell connectivity. And if you look at any of the classic angiogenesis assays, they all use fairly soft materials, collagen, fibrin, matrigel, things like this. All of the work that Judah Folkman did and basically the field he created was based on these soft substrates that we know can foster angiogenesis. So based on our data showing that there was this differences in connectivity, we asked about barrier integrity. And so we took our endothelial cells, we plated them on hydrogels of different stiffness, and we simply looked at how easily dye can move between monolayers of cells, across a monolayer of cell into the hydrogel. And we found as the stiffness of the material increases, the permeability of the monolayer goes up. So again, this is not in the presence of thrombin or any sort of agonist. This is simply the stiffness of the material, and we see an increase in the permeability. Now, we did a lot to work on that problem, but one of the issues with the material is that they're all 2D. The best way to control stiffness generally is a 2D material, but cells, of course, don't exist in 2 dimensions. And so we started looking at materials that we can manipulate the mechanics of in 3 dimensions. Most recently, we've been using these materials. Basically, it's a collagen that's soaked in high concentrations of sugars. The sugars glycate the collagen. This is to cross-link it, and they can produce matrices that are increasingly stiff, but have a very similar architecture. So you can see here, this is collagen and 0 millimolars of ribose. This is 100. The structure looks very similar, but the stiffness of it goes up about 2.5-fold. So not as big a range at all as compared to what we can make with, say, a polyacrylamide gel, but we can still get a change in stiffness. And what we find is that if we take endothelial cell spheroids and we put them into these glycated collagen matrices, as you increase the extent of glycation, so that's going down, you increase the amount of outgrowth. So we quantified that. You get more longer outgrowths, and you get more of them, and they are also longer in stiffer, more glycated matrices. This is just a pretty picture showing that over the same time interval. In a soft collagen matrix, you get some outgrowth, but with a stiffer, more cross-linked matrix, you get significantly more outgrowths. What's interesting, you don't just get more outgrowths, you also get more branched outgrowths. This is us quantifying branch density. The reason we found this interesting is because branching is a hallmark of tumor angiogenesis. Increased branching, increased tortuosity. And we've shown here that at least partly cross-linking in the matrix can control branch density. I'm going to speed through this. One thing we did was everything I just showed you was changing stiffness by changing cross-link density. But if you look in the literature, basically right before we started publishing, people were basically coming out with the opposite relationship. So our data that I've just shown you is basically that stiffness increases angiogenesis, but it's stiffness by cross-linking. If instead you change stiffness by changing density, you make increasingly dense collagen gels, you get the opposite relationship. Stiffness decreases angiogenesis. And we see the same thing in our system. So for any glycation, any amount of glycation, you get increased angiogenesis. But then if you look from a really low density collagen gel to a high density collagen gel, you get less angiogenesis. So what does this tell us? It's just important to qualify what is controlling your changes in stiffness, whether it's density or cross-linking. So we did, so all those experiments I showed you were all in vitro in collagen and Petri dishes. In these experiments, we took our collagen and we put them into a CAM assay. It's a very commonly used angiogenesis assay. Basically, you put it on top of a developing chick embryo and you can watch the blood vessels grow in. And what we find is that we get increased vessel density in our stiff collagen gels compared to our soft collagen gels. And we also moved it in vivo. So here we use the BAPN mouse. So this is a mouse, the MMTV PYMT mouse that spontaneously develops breast tumors. If you feed it BAPN, the tumors still develop, but they're quite soft compared to the control tumors. And what we find is that we get significantly less blood vessel growth in our soft BAPN-treated tumors compared to our stiff control tumors. And this is us quantifying that. We get less branches and less junctions, so less total outgrowth when the tumors are softer. Okay, so what about, so that's vessel number and vessel branching. What about vessel integrity? So we did the same experiments in mice with our BAPN mouse. But we used a FTSE dextran to look at permeability of the blood vessels. So we inject dextran, which is going to tell us about the permeability, and we inject Evans blue, which is going to tell us about the architecture. And so these are histology sections from those tumors. And what you can see is that in our control tumor, our stiff control tumor, we get much more permeable blood vessels. In our softer tumor, we still get vessels to grow, but they're much more firm, better formed with better integrity. Okay, so I'm going to speed through this in the interest of time. And I'm going to basically just tell you about our last piece here, trying to tie it together. So basically what I've shown you is that stiffening through cross-linking increases branching. And the natural question is why? Why is that happening? Why is there increased branching? And going back to our other data, looking at looking at collective migration and looking at the energetics required of leader cells, the question we're now asking is whether or not the branching is actually related to the energetics. So in other words, as the cell is moving in a stiffer matrix collectively out to form these angiogenic sprouts, the leader cell is burning a lot of energy. So this is our Percival probe. In our high energetic state, you can see significant energy burn at our tips, and asking whether or not that's related to branching. So this is a major avenue of research that we're pursuing now in the lab, trying to connect what we've shown in collective migration in cancer to collective migration in angiogenesis. With that, I'll summarize. Basically, what we've shown is matrix stiffening can disrupt vascular growth and structure. It's mediated by matrix mechanics. What I didn't get to is that they're not limited to endothelial cells. So these problems in cell-cell connectivity extend to many cell types. Epithelial cells in the gut is an example, skin cells, basically anything that's going to have a balance between cell-cell and cell matrix interactions can be disrupted based on matrix stiffness. And then last but not least, some of you may be aware there's emails flying around right now talking about mechanomedicine and the origin of mechanomedicine. My lab wrote a review on mechanomedicine a couple years ago. I'll really push most of the credit to this fabulous graduate student I had, Marsha Lampe, who went into the literature and dug out clinical trials that were in place that were basically going after mechanical targets. So basically everything she could find in the pipeline that could be promising, looking at molecules that are mechanosensitive. Again, it sounds like I'm complimenting myself, but it's really the student. She did an amazing job of really pulling together some of these fundamental findings that those of us in mechanobiology are looking at and trying to connect it really clearly to medicine. Okay, acknowledgements, the folks doing this work. So Matt Zanatelli did a lot of the ATP work on single cells and microchannels. Jan Zhang's a really talented postdoc in my lab who did a lot of the collective migration work. And then a lot of the angiogenesis work was actually my former postdoc, Francois Bordelot, who's an independent faculty member now at University of Laval. And with that, I'll take any questions.
Okay, great. Thank you so much. Let me just figure out how to remove your spotlight. Okay, great. So we have some questions in the chat, and just to remind everybody, please go ahead and either write them out in the chat or feel free to ask the question yourself. Just let us know you have one and we will call on you. So the first question is, does glycation change interactions of the cells with the collagen fibers at all?
That's a really important question. So I'll be honest, when we first started this, we didn't really want to think about that. We just liked having a tunable system. But the fact is, glycation is a chemical process. It produces crosslinks and it produces something called advanced glycation end products. Glycation occurs naturally as well. It occurs with age. It occurs more rapidly in diabetics. And these advanced glycation end products, there's actually a receptor on cells called, those are called AGEs, advanced glycation end products, and the receptors are called RAGEs, receptors against advanced glycation end products. And most cells do have receptors for RAGEs. So I didn't present it here, but for a lot of the angiogenesis work, we had to spend a ton of money on RAGE blocking antibodies. And actually we're repeating some of that work now CRISPRing out the RAGE receptors, so we can get rid of those effects. But yeah, glycation can have an effect independently of stiffness.
Thanks. I have a quick question, which is in the assay where you showed the switching of positions between the malignant and non-malignant cells. So when the non-malignant cells become the new leader, are they actively leading and increasing their metabolic output and contributing to the outward growth, or is it just sitting there as a leader until another malignant cell gets there?
That's a good question. So at least in our hands, we looked at pauses. So we looked at like the strand outgrowth to see if it, if it pauses or if it continues to grow. And as the switching happens, there's a pause. But after the switching happens, it basically continues to grow. The amount of data we need to determine whether or not the rate is exactly the same, we haven't gotten to yet. But it seems to take on the role of leader. And I'll tell you, the data is, in full disclosure, the data is somewhat controversial. So there's some good work by Adam Marcus's group in this area, looking at leader-follower behavior, and they don't report switching. They report it, but on very, very long timescales. And so it's generally not really, part of the question they're asking. Most people view the leader as the leader. What are the traits that make a leader cell a leader? A lot of Andy Ewald's work identifying specific markers of leader cells has been incredibly important, but also suggests that there is a leader cell. But if you look closely, a lot of that work, it's not usually just the first cell that has a lot of those markers. It's usually the first few cells. And so we think that there's usually a few cells that either take on that role or are already equipped to take on that role. And that's not clear to us yet, but we're working on it.
Great, thanks. Our next question is from Ashri, who says, very interesting talk. And then in the first part of the talk, you show a connection between metabolism and cell migration, but there's also a lot of literature connecting actomyosin and cell migration. So is there a scaling between cell metabolism, actomyosin, and cell migration?
That is a great question. We have not looked at that. I bet there is, though. It would just make sense. And I think most people, what they would tell you is whether or not you can pull out the scaling because, again, of this really prominent role, a lot of the other things that the cell is doing in metabolism. So I bet it does scale. What I don't know is whether or not you'll be able to detect that scaling behind the noise of everything else that's metabolically active in a cell. But it's interesting. I'd have to think about how to get at it if that were the case.
Thanks. Oscar Ruiz says, great talk. I'm curious about your thoughts on the role that chemokines may play in a cell's decision-making when looking at different size spaces.
That's a great question. So actually, a few years ago, I tried writing a grant to get at that, chemokines and hypoxic environments. So what if you could take one channel and create a gradient of chemokines or create a hypoxic environment in one versus another, and how that affects the decision-making path. And what I realized in trying to write the grant and design experiments was I hit my wall in my micro device abilities. So I bet somebody on this call could probably get a good device to be able to do it. One of our stopping points one of our hurdles for this is that we feel pretty strongly about the use of collagen for these channels. There's a lot of groups that use PDMS and have gotten some really great results. Kostas Konstantopoulos comes to mind, Sanjay Kumar, really helping to define how cells perceive confined environments using PDMS. We use collagen because we think it better mimics the tumor microenvironment. But it does pose a lot of fabrication challenges because things like creating a gradient in something that's highly diffusive and highly porous is, technically very challenging compared to solid PDMS walls. So I think they do play a very important role. I think you could drive really large changes in ATP using things like oxygen, for instance, or glucose gradients. Which I think are probably very physiologically important, but we don't have a good platform yet to figure it out.
Oops, great. Hi, sorry, had some mute troubles. From Raphael Petrosian, thanks for the interesting talk. For the cells that were touching both walls of the path, they're mostly choosing the narrow path when given a choice. And what's the principle behind that?
Maybe I didn't represent that data well. So if they hit the bifurcation and they're touching they're touching both sides of the feeder track as they hit the bifurcation, they tend to choose the wider track, not the narrower track. And we think that that is energetics. If I didn't show you time lapses, but if I had, often what we see is cells will send up extensions into both and then make the choice. So we got snagged on this when we were trying to publish this paper because the reviewers didn't want us to sound like the cells were sentient beings, which I can appreciate because they're not. But there was definitely, it wasn't just a simple case of like how water flows through a pipe of choosing the wider path. It was most definitely a case where a cell would hit a bifurcation, it would sit there and probe up each side, and then it would make a decision.
Great, thank you.
Keith Storm says, fantastic talk. In the cell drafting, how important is actual mechanical pulling via cell-cell adhesions of followers by the leader? Would the leader just be the fastest cell dragging along the rest?
That's a great question. So there's the really good work out of Eric Sahai and Javi Tripat looking at this and the idea that the leader cell can connect and pull. In our system, I mean, there are cell connections, but there are also not cell connections. So we can find cases where the leader is just a micron or two ahead and we don't see any direct connection there, where pulling is actively happening. I have no doubt that pulling happens in systems. We don't see it in ours. Because again, we can see at times where they aren't even directly in contact and we can identify this behavior. We've also done the work using organoids. So taking tumors, digesting them just a little bit and putting small bits into collagen. And even in those cases, we can find cells certainly that look like they're pulling, but we can also find cases where the leader cell is not directly in contact with the followers, and the followers are still following behind. So is the leader cell the fastest dragging cell? It's a good question. We've also done experiments that look a lot like Adam Marcus's, where you can photoactivate the leader and then sort them out. And it's a pretty robust population of cells. So you can sort out leaders, put them back into a mixed population, and they remain leaders. And I didn't show any of this work here, but we do have data to show that the migratory phenotype of a cell is fairly stable. Meaning if you were to sort, you can sort cells based on how migratory they are out of a parent population, you can sort them and they can remain stable in that phenotype. So the idea that they could just be the fastest cell is entirely possible, but I'm not sure that it's that they're dragging, but they could be that the fastest cell that provides the leader behavior.
Great, thanks. From Kostov Bera, very interesting talk. So related to the single cell work and the first part, did you see any phenotypic changes for the cells with higher levels of ATP besides the path selection, i.e. morphology, invasiveness, proliferation, et cetera?
Oh, that's a good question. So we have not done that, mostly because the way we did those experiments is we went back and after the fact, looked to see what their ATP levels were. Meaning we measured their ATP levels, we identified which path that we took, and then we went back and said, okay, you took the wide path and we're gonna lump you into this category based on your behavior before that. So we didn't really sort them based on, we didn't have two subpopulations, but we are working on that now. So we're working on, the photoactivatable probes are just awesome because basically it's allowing us for any population that's stable, where you can maintain a phenotype after proliferation. It's a really, assuming that works, which isn't always the case, it allows you to sort cells based on all sorts of things, including ATP. So that's something that we're working on now.
Jeff, do you want to go ahead?
Oh, my turn?
Yeah.
Okay, yeah, this is Jeff Frederick. Thanks, Cynthia. I really enjoyed that. You've really pushed the field a lot. My question is that two papers appeared just in the last couple of months. One is from Peter Friedel's group in Nature Cell Biology that really showed that it's the way the cells move collectively in collagen channels is all about jamming and unjamming, and the role of crowding and confinement. So I wonder if you'd comment on that. And then we had I mean, we're not nearly as sophisticated as you in terms of metabolism, but we showed that jam cells are more economically, energetically, and unjammed cells are less economical, they're expensive. So rather than drafting as the model, maybe is jamming. How do you see jamming and unjamming fitting into this picture for collective behavior?
I think that's a great question. So I don't think the two models are necessarily mutually exclusive. I think that there's data to suggest that you could have both an jamming and pushing phenomenon at the same time that you have leading. So I'm not sure. I'm not sure which one dominates. But I think I saw, I don't know if your papers come out, but I think I saw it on bioarchives of looking at metabolism and jamming, unjamming. And I think I mean, it's just amazing work. I mean, I think it makes a lot of sense to me that it plays a role. But again, in terms of which one dominates, I'm not entirely sure. I think the other element to think about too that we've been giving some thought, and I know Peter Friedel's group has been giving thought to as well, is which mode the cells are using to make energetics. So clear differences between use of glycolysis versus oxidative phosphorylation in leader versus follower cells. Our data right now doesn't really mesh with some of the other stuff that has come out specifically from like Adam Marcus's group. So we're still trying to figure out why that's the case. But I think, you know, at least part of it is that you're going to see differences in not just how much energy it takes to do something, but how the cells are going to generate energy to do it. But I don't know. I guess I could ask you too. Do you think they're mutually exclusive in terms of the ability of cells to lead and basically carve paths as opposed to jam and unjam to move collectively?
I mean, I would say they're not only mutually exclusive. I think they may be the same thing. I think they're connected to each other. Yet we know that the leader cells do pull more. They create greater forces. They're going faster, and they do change places with the ones behind them. So, but they're also unjamming. So, I think they're just different views of the same phenomena. So, yeah, totally not mutually exclusive. I think they're probably intimately related.
I think so.
Yeah.
Okay, great. Thanks for that discussion. Cindy, are you okay to stick around for a few more minutes? We've got a few more questions on Zoom and then a few from YouTube as well.
Sure.
Okay, great. So two from Anna Pasapera. Very interesting talk. For the first part, do you see differential activation of AMPK during migration?
That's a good question. So we haven't looked at that. We haven't looked at AMPK activation, but we have tried to manipulate AMPK activation to see if we can drive differences in behavior. And honestly, the data is all over the place right now, so I can't even really tell you what it is that we're finding because I don't know. What I did learn. So we're new to metabolism in that it wasn't something that we started thinking about until about 4 years ago. And so thinking about it now in the context of the actual signaling pathways, what we've learned is something like AMPK, you know, it's this master regulator of cell metabolism, but at least to me, it's not necessarily intuitive how it works. So in some cases, it actually has, well, for lack of a better word, counterintuitive effects on the metabolic activity of cells. So I think that's a pretty open-ended question. I think it's an important one to get at because, again, AMPK is this master regulator. And I think we are going to see differential activation based on the position in the leader versus follower. You need more energy from the leader. You're going to need to activate more of the metabolic pathways.
Okay, great. And the second question from Anna Pasapera, does matrix stiffness regulate VEGF expression or VEGF receptor activation?
Oh, that's a good question. So we actually had a paper on this a couple of years ago, And it was a hard paper. I thought it was going to be very easy just to look at synergistic regulation of VEGF activation on soft versus stiff substrates. But it was not. We do see differential activation, but not because of expression levels. What we found was that there were differences in the rate of endocytosis of the VEGF receptor. So VEGF receptor recycling on varying stiffness matrices. And other people had shown this in the context of mostly like drug delivery, like uptake of drug delivery particles and matrix stiffness and things like this. But at the time, we didn't know of anybody who had looked at it with in the context of growth factor receptors. But that's what our data suggests, that yes, there is differential activation of VEGF receptor, but it's not due to expression, it's due to basically endosomal sorting.
Thanks. Question from Padmanaban P. Great talk. You mentioned cells draft. What's the relation to cells in terms of energy when external fluid flows or chemical gradients was applied?
Good question. So we haven't tested that. So I mentioned the chemical gradients. It's something we'd love to do, but we need a better platform to do it. In terms of external fluid flows, so in the case of like collective migration for cell drafting, the only external fluid flow that'd be really important there would be like interstitial flows, like due to like lymph system and things like this. It may be really minor flows. I'm not sure whether or not they would have a role. I don't feel like I have a good physical intuition for that. In the context of angiogenesis and external fluid flows, again, the timing of when fluid starts to flow in the newly forming vessels really has to do with when they actually form a loop, in which case I think you're past that point of tip dynamics and things like that. So I'm not sure that external fluid flows are going to have a big effect there, but maybe there's a system I'm not thinking of.
Okay, great. From Samantha Stam, thank you for the great talk. Related to the first part, do cells dynamically change their ATP-ADP ratio in response to a barrier or a change in the channel width?
They do. So we have some work we haven't published where we basically created a three-dimensional interface of collagen. So basically 2 collagen blocks up to each other, where a collective strand basically hits the interface. And it does one of 2 things. Either it steers away from the interface, or if it goes through it, you can see a notable increase in the ATP as it moves into the denser matrix. And same thing with confinement. If you watched a cell as it moves from a wide channel into a narrow channel, or even down a tapered track, you can watch the energetics go up. And so, one thing we're not clear on is how that's regulated. So how does the confinement lead to active changes in cellular energetics? We're not sure, but most definitely cells will basically up their ATP production. We tried to figure this out. I mean, it must be known, and we haven't found it in the literature, but the idea that basically cells make the energy they need. They don't typically store a lot of it, or at least not in our hands, they don't. And people have suggested that they just make it as they need it. But we've had trouble finding that reported rigorously in the literature. And again, I feel like something we're just missing, because one would assume one would know that.
We've got a question from Yaboa Amofa. Thanks for the great talk. Do you think that leader cell switching might be cell or cancer cell types dependent?
Oh, I think so. And that's something we're looking at. Now, I want to be careful, because I think there's a fallback for a lot of people. Anytime 2 people's data don't agree, they say, oh, we're using a different cell type. So I want to be careful not to just to fall onto that. But I do think that dynamic behavior is probably cancer type specific. We've looked at several, and we're expanding now. So what I talked to you about was all in breast cancer, and we have some work now looking at GI cancers, colon cancer, Skin cancer is also, melanomas, well known to migrate collectively. So a lot of our work is springboarding from the systems where people have already identified collective movements as important. But certainly I think there are exceptions to that.
I was just thinking, have you seen the new paper from the Denuser lab? They talk about they call it worrying. And I haven't read this, I just saw people talk about it on Twitter. But I think basically the idea is that for invasion, that you can get these cells that are just sort of, they're sending out, protrusions that are just kind of like scooching the matrix out of the way, as opposed to doing any sort of degradation or anything, sort of making paths by just sort of continuously doing little pulses and little pushes. I was just wondering if you think that that fits into the picture that you have of, the metabolically active cells in the front. Maybe those are the ones that are, doing this pushing.
So actually, I think my student may have just sent me that paper last night, and so I haven't looked at it yet. And it's something like your timing for the question is amazing, because literally just yesterday I was talking to my student about this, that the role of different protrusions and which protrusions can actually exert forces and which ones don't, because as you know, if you look historically back, it seems like protrusions keep getting defined and redefined. And the idea that, originally Philopodia weren't supposed to be able to do anything. They weren't supposed to be able to do anything but feel. They can't really exert forces or things like that. There's a paper earlier this year showing they most definitely can exert forces and move fibers. So, how big of an extension does there need to be, and how much remodeling can it actually do in helping a cell move? I didn't talk about it all here, but we have sorted out a subpopulation of cells that do exactly what you described. So they come out of a parent population that's highly migratory, but we sorted for their ability to not migrate. And they do exactly that, a lot of like frustrated protrusions where they just sort of keep sending them out. They look like they have all of the mechanics necessary to be able to move. They are very active. They're metabolically active. They can secrete MMPs. They clearly have a ton of cytoskeleton, but they don't move. And so, why is that? Or they move very little, as you suggest, they just sort of push until they wiggle along. And why is that? Probably has to do with polarization mechanisms, but we haven't looked at it.
Sukanya Gayan says, with the spheroid experiments, did you check how different layers in the spheroid behave metabolically different?
Oh, that's a good question. So one thing that we've done is we design our spheroids to avoid necrotic cores. So we make them small enough that we don't think that much recently, but in the past, is this differential adhesion hypothesis. So the idea that, in a collection of cells, you're going to get really highly tightly bound cells surrounded by less tightly bound cells, surrounded by less tightly bound cells. And that is one of the theories of how cells sort during development. And people have used it to explain how tissues spread and things like this. But if we were to take that concept and look at it in the context of energetics, absolutely, I think there's a difference. And this goes back to Jeff's question about jamming and unjamming in those systems, and the cells basically shifting around and pushing outward. I think you'd most definitely see a difference in the metabolic activity. We haven't looked at it. I don't know anybody that has, but these probes would be a great system to do it.
Okay, great. I think we're down to the last two questions from Gijje. I'm sorry for the mispronunciation of your name. Have you looked at the spatial localization of mitochondria in invading cells, for instance, and then explored leaders versus followers?
So there is some really great work coming out on that that was published a couple of years ago, and it makes perfect sense to me, but we haven't been able to see it in our system. That we would have thought that we'd see mitochondria differentially localized to the leading edge of the cells, but we don't see it. But what we do see, which I didn't discuss at all here, we've done separate work with CAFs, cancer-associated fibroblasts, because there's some data to suggest that they can serve as leaders and that they can pull strands and things like this. And we started doing those experiments just to put them in the context of our leader-follower behavior. But what we accidentally discovered as we were doing those experiments is that The cancer cells can actually take on mitochondria from the calves. And when they do, they become much more metabolically active and much more migratory. But the mitochondria don't seem to differentially localize in the cell to the areas of most activity, not in our hands at least. So we fully believe the data. It makes 110% sense to us. We just haven't been able to see it in our system.
Thanks. And the final question today from Rupa Raman Mishra. I was curious whether the response to stiffness of angiogenesis would also correlate to the type of cancer, given the response to stiffness depends based on the type of cancer.
So, so the question is whether or not the angiogenesis response to stiffness is cancer type specific?
I think so. Maybe if you can talk about, yeah, the response of cancers and angiogenesis to different types of stiffness, I think.
Well, so I guess that there is an important point here, which is that, you know, some tumors just in general are much more vascularized than others. Whether or not that's related to stiffness is an interesting question. I haven't thought about it. I mean, I think there's just some tumors that, you know, clinically they focus much more on the vascularization because the tumors just are more vascularized. But whether or not that's a function of stiffness or extracellular matrix, we have not looked at, but it's definitely an interesting question. And I don't think I know enough to speculate, but I think it would be something worth looking at. And actually, it reminds me, when I very first started working in this angiogenesis, the question of tumor angiogenesis, I presented some of our really early work at a conference that Rakesh Jain was at. And if you don't know Rakesh Jain's work, I mean, he's basically a pioneer in the concept of tumor angiogenesis and mechanical forces affecting it, and the idea of normalizing tumor vessels and things like this. And it was actually a suggestion that he had to me to look at different vascular beds, and we just didn't pursue it, mostly because at the time I didn't have the resources to do it. But I think it's definitely an interesting question.
Great, thank you very much. I think we are all done with questions finally.
Okay, great. I'll go ahead and stop the YouTube live, but just want to say thanks again to Cindy for that wonderful talk and for sticking around to answer all the questions. Thanks for everybody for being here, and we will see you guys next week.